So as you might have noticed, I've changed the title of my talk just a little bit from what my abstract is, in part because I think this will be more interesting. But if you really want to talk to me about the trials and tribulations of using multi-TOF mass spec later, I'd be happy to take those offline. So as we went over this morning in our morning talk, I think we're all acquainted now with the structure of RNA. So again, we have those ribose sugars, phosphates, and nucleobases. And all of these are connected via phosphodiester backbone. What I'm most interested in are these nucleobases. So we know that we have 4 that are included in modern RNA. And that includes 2 purines, which are adenine and guanine, and 2 pyrimidines, which are uracil and cytidine. And we also know that the purines will base pair via hydrogen bonds to the respective purines. So again, A to U, G to C. What's most interesting about these nucleobases is that there could have been a whole library of possibilities to be included in our genomic DNA and RNA. Yet we only see 4 included in modern RNA. So what I'm most interested in is trying to determine how we ended up with just these 4. And most specifically, I'm interested in guanine. So guanine is relatively unique compared to the other modern nucleobases. And this is because guanine can actually base pair with itself via hydrogen bonds in very concentrated solutions. So if we have a concentrated solution of guanine or guanosine monomers, we can get these structures of G tetrads that are, again, hydrogen bonding to each other, very much like we see in base pairs. And if we increase this concentration of guanine even further, we can get even larger aggregate structures of these G tetrads. So these G tetrads are just planar arrangements of the guanines. These G quadruplexes are just a G tetrad stabilized by a metal cation, typically potassium sodium, and then another layer of G tetrad, metal cation, G tetrad, et cetera, et cetera. And as far as we can tell, guanine is the only one is really the only nucleobase that we see in modern RNA that really does this, or at least to any extreme extent. So this brings us to why G. So if G can form these large aggregate systems that are much easier to assemble than having it go into RNA, then why is it included in RNA when it can do this? So it could be in spite of this ability to form these aggregate structures, but we like to think in our group that's because of this. So this leads to what my work is, which is looking at the effect of GMP, or guanosine monophosphate, on abiotic polymerization. So this means that I'm looking at the inclusion of other nucleotides based on the presence of GMP or sequence effects that are caused by constraints caused by these G aggregate structures. What the focus of my talk today is, though, is just looking at the inclusion of these other nucleotides in our synthesis reaction. So to make polymers from various unactivated species, it's impossible. So we need some sort of catalytic reaction to happen. And so we use catalytic Montmorillonite clay. So this is clay that you've already heard about several times now, that are just layers of tetrahedral and octahedral arrangements of atoms with this space in between that can hold cations. And we've seen there's been some previous work, especially done by the Ferris group at RPI, that shows that you can get RNA molecules both inside and around the edges of the clay, and that this will lead to polymerization of activated nucleotides to as long as 50 bases long. But again, this clay is not enough for our reaction to happen, so we also have to have an activated species. So if we think of some nucleoside monophosphate like GMP, CMP, AMP, UMP, this hydroxyl group is not a strong enough leaving group to allow it to polymerize into long strands of RNA. So what we do is we exchange out one of these hydroxyl groups for an imidazole right here. And this makes for a very good leaving group in water systems, which makes it significantly easier to create these long strands of polymers. To analyze my polymers, I use something called matrix-assisted laser desorption ionization, or MALDI, time of flight, TOF, mass spectrometry, MS. And in short, All I'm really doing is hitting my sample with a laser, causing it to ionize, and then I have it flies through a long tube, and it separates based on its mass to charge ratio, which you can see determine via kinetics equations and look at how long it takes to fly through the tube. So in short, what I get are spectra that look like just a separation of peaks, and I'm identifying my polymers based upon the separations between those peaks. So again, I need to put my, activated species in clay, and I can get a polymerization in the case of IMPA, so activated adenosine monophosphate, to about somewhere between 8 to 12 mers, which means 8 to 12 units of A. So this one up here shows an 11 mer, up to 11 mer, which is pretty cool. And if we add in any unactivated species, we can see that we get still a similar amount of polymerization, a little bit less, but it still kind of works. We're still getting a decent amount of polymer formation. But if you were listening to what I was talking about earlier, you might expect that we'd only get one peak. But if you look even closer, you'll see that we get multiple peaks for one species. So if we zoom in on just one set of peaks, so I'll zoom in on a 5A, we can see that we do get several peaks. So our base peak is a 1663 in the case of 5A. So again, this corresponds to a polymer of A that has 5 adenosine monophosphate units in it. We can see that we get some peak separation that's like plus 22 for a sodium RNA aggregate, plus 38 for a potassium RNA aggregate, and a few other species as well. But what's most interesting is not these aggregates with salts to us, but if you look at the addition of unactivated species, CMP, AMP, UMP, they look fairly similar to just IMPA alone in clay. But GMP pops up with this other random peak at that is plus 16 difference from the 1663 peak. And that plus 16 corresponds to the addition of a G instead of an A. So what that means is that this peak here is 4A and 1G. So again, a polymer of RNA, 4A base units, and 1G. So this is really cool. We see that GMP seems to force itself into polymer inclusion, whereas the rest of these don't. So of course, one of the next things we want to do is look at an entirely different system. Well, not entirely, but fairly similar system. So of course, we look at G. So this here at the top is our IMP-G polymerization in clay. So again, you can see that we get a little bit less polymerization than we do for A, but we still get up to 7, which isn't bad. If we add in the unactivated species, we get fairly similar extensive reaction. But again, we're getting multiple peaks where we might only expect to find one. So if we look again much more closely, we'll see the sodium peak, we'll see potassium and stuff. If we look at the IMP-G polymerization, we see that we have a 3G here at 1052. And if we look at the one that includes GMP, it looks fairly similar, a little bit of extra baseline noise, but essentially the same as our IMP-G reaction up top. But the minute we look at the CMP, we notice an entirely different peak that's minus 40 mass units from our base peak of 1052. And if you want to math it out, you'll notice that C is different in mass from G by minus 40. So this suggests that we are getting a different type of polymer here that is 2G and 1C. And if we look again at A, we're getting a similar thing where we're getting minus 16 for 2G and 1A. And we can even make an argument down here for the U, where we're getting, again, minus 40 because U and C are approximately the same mass for 2G and 1U. So this is really cool that we're getting, in our abiotic polymerization that's using an activated nucleotide and unactivated species, we're getting inclusion of these unactivated species in our polymers. But this still isn't enough. So we've recently tried to get some preliminary data on the polymerization of C. And unfortunately, this is only a first run, so our C did not polymerize quite to the extent that we had hoped for. But what's most important is that if we look at the inclusion of low concentrations and high concentrations of GMP into these reactions, we can see that we are getting peaks that correspond to, again, the addition of a G in place of a C in our polymerization reactions. So it does appear that G likes to force itself into our other polymerizations. So some quick conclusions. Whenever we make our poly A from our IMPA and clay reactions, we get a G incorporated in, while no other nucleotide likes to incorporate itself into our polymers. If we look at our poly G reaction, we'll see the incorporation of all the unactivated species of nucleotides. And we even have some evidence that suggests that G will incorporate itself into the C polymerization. So all this data together is starting to suggest to us that maybe G is including itself because of its weird properties, its unique properties, and helping to bring others in addition to these, because of these unique properties. So obviously some future work, we want to make our IMP-C reaction look better, and additionally we want to get IMP-U to get polymers using our IMP-U reaction, and also looking at using unactivated species as well. Excuse me. And eventually we do want to get to quantitation and sequencing of the species that we're making, because MALDI-TOF can't quantify or sequence our species. We can only determine the general contents of what we have. So I'd like to finish up with some quick acknowledgements. So thanks to my advisor, Dr. Linda McGowan. Of course, thanks to my group, especially Bradley Burkar, who gave a talk yesterday, and Lauren Cassidy. Thanks to the Ferris group downstairs from us at RPI, especially Dr. Prakash Joshi, my committee members, and of course I do all my work at RPI, which is one of the NASA Centers for Astrobiology, and I'd be happy to take some questions.
Thanks for not making me go to.
Interesting. What is the sort of the mechanism that you're seeing? Because it seems like there could be two competing processes. One is going into the tetrad formation, and then the other is actually being absorbed to the Montmorillonite. And that seems like they might be mutually exclusive. Do you have any idea?
I'm not exactly certain what things are going into the clay and what are not. Some data that I haven't showed suggests that all of our unactivated species do get absorbed into the clay at some point, or at least in varying concentrations. And so I would guess that a decent amount is going into the clay, allowing us to do our polymerization. Does that make sense?
Do you know if you're still forming tetrads?
At most of these concentrations, if we do get some tetrad formation, it's very low. So it's possible, but it shouldn't be having as much of an effect. In fact, the IMP-C, if I can go back. So I was trying to get higher concentrations of GMP to force that quadruplex and tetrad formation. But we can look at it, and it doesn't look like it had too much of a difference in effect. Maybe slightly lower polymerization and more aggregation instead.
Any other questions for Kristin?
So, because we both do this, I have a very specific question for you. Okay. So, can you actually go back one more slide? So you used your IMG and you used the 4 canonical nucleobases as inactivated species, and because you're trying to figure out why we use those 4 and not other nucleobases, have you considered using the non-canonical nucleobases as the inactive species? I know they're expensive, but have you?
That's a beautiful question, and actually that's a long-term project.
Okay.
So in the work that I'm doing, hopefully by next year I'll start this, is starting to include some of the other purines that we find, especially in meteorites, so like xanthine, hypoxanthine, purine, and some of the diamino purines. Mm-hmm. I have not yet done it, but I'm really excited to do it.
Okay, because we can go halvesies on some of the expensive ones if you want.
All right, let's thank Kristin again.